If you could optimize the genomes of underexplored microorganisms such as filamentous fungi, you could significantly broaden the biomanufacturing toolbox, claims Anthology.
When we think about like biomanufacturing right now, it's usually limited to, say, you begin with E. coli and yeast, so you're naturally limited by their capacity of doing things. So while we think about like how we approach biomanufacturing or how enabling our technology is, it's going to be like this. So in 5 to 10 years or even shorter, you have a feedstock you really want to get rid of. For example, it can be egg waste, can be animal feces, can be plastics even, and you have a product that you really want to make. Can be materials, textile, or high-value proteins. What's connecting the two nodes is going to be a genome, an organism, and we want to generate that organism using a combination of genetic engineering, hardware engineering, and AI tools. So we are not bound by species anymore. It can be anything. Can be a hybrid, can be a chimera, can be a mutant from a very closely related mushroom. Yeah.
While other companies are trying to engineer cells like computers, Anthology takes an entirely different approach by learning from what nature does the best. So using our technology that combines innovations from genome engineering, hardware engineering, and computational biology, we are making a platform that basically accelerates evolution by speeding up the process of generating genome mutations and then also picking the winners from our custom build devices. And then essentially this allows us to generate a very large data set that maps all these genotypes to downstream phenotypes, basically enabling the future of generative genome design where given any feedstock and any product of interest, we can design a genome from scratch. We love fungi because they're so hard to work with. Yeah, so basically E. coli, working with E. coli, you can see the results within 1 week, and then similarly yeast, you can see the results within 1 to 2 weeks. But for fungi, each engineering cycle takes up to 10 weeks until we see a known phenotype. Yeah, and then we really have to be very creative in the way how we address this engineering problem by trying to be multiplex and then trying to generate a lot of genome variants in a short amount of time, and then use microfluidics devices or some other high throughput equipments to screen the candidates that are best fit for certain purposes. And then we are also recently starting to decoding the language of protein secretion of fungi by applying AI tools. So we are trying to learn the structural relevance for protein production, and then eventually that will allow us to generate a panel of different hosts that can make different type of proteins of interest.
How do companies currently try and optimize their strains?
Basically, there are two mainstream approaches about doing this. So the first one, like you just described, you can imagine the cells as a computer, which by now we know is probably not the case. But anyway, you can guess there are like 20 different control knobs. You can tune them one by one. But as you can imagine, this requires a lot of workforce, a lot of money, and of course time. And there's another approach which has been around for a while. This is like people trying to imitate evolution. But we have very limited amount of tools that we can do that. So usually people use toxic chemicals or UV to introduce mutations across the genome. So this is a little bit like playing lottery with extremely low odds.
So in layman's terms, what are you doing differently?
We use genetic tools so we can actually change the order of paragraphs. We can change the order of chapters. We can remove a few pages. We can even borrow a few pages from another book. So by doing this, we can really introduce diversity across different scales. And most importantly, if you see some changes in the phenotype, meaning the traits you like, you can go back to the book, looking at it through DNA sequencing, and figure out what kind of traits is caused by what kind of mutation, and eventually you can use that to learn the language of life, or like stacking different traits together.
So you're kind of introducing these genome mutations at a really grand scale, and then how are you kind of screening to see which mutation leads to the better phenotype?
Evolution is really good at doing a lot of different kind of things, so it depends on the traits you're interested in. So if you are thinking about, for example, the ability to use waste as a feedstock, you can let them just compete against each other. So millions of them in a bioreactor is like running a marathon. You only care about which ones are the fastest. But there are some traits that cannot be analyzed this way. For example, if you have a group of singers, you want to analyze their ability to sing, you cannot really let them to sing in the small room. and then at the same time evaluate them individually. So what you do is like put them into different rooms. So that's what we do with the cells. We put them in different droplets or beads, and we pass them through a channel. So at any given time when they pass through the channel, we analyze them using lasers or other optical tools. So we know what they look like, the amount of protein they are secreting, and we can do real-time scoring and ranking, and we can sort them. And then from there, those winners, we can study their genotypes, or we can put them through a pipeline of scaling up and validation.
So you're building on this Nobel Prize winning work from Barbara McClintock on so-called jumping genes. She was looking at different generations of corn plants and found that instead of genetics being kind of locked into place and giving these fixed instructions generation to generation, some genes can jump around and kind of transpose. Can you translate, her work into what you guys are doing?
So what we do in the lab is we actually take mobile genetic elements or like jumping DNAs from like a distant host. And then from there, we use a lot of modern genetic tools to add their functionality and most importantly, controllability. So we can make sure we can precisely tune the jumping event in our host of interest.
Currently, we're pressuring test different type of business models. So like you mentioned, there are two approaches. One is to prove the technology in existing industrial hosts, like showing that using our technology, we can improve the cost margin for our partners. So we have demonstrated some success over there. And then at the same time, we're also exploring unmet market needs, like talking to large industry partners to see what proteins they want to produce or what feedstock they want to utilize. So from those conversations, we have generated some high conviction protein targets, and so far we are negotiating with larger commercial players to be able to produce these proteins and then push them towards the finish line. The whole company was built on the basis that we are addressing a real industry need instead of building a set of cool technologies.